Okay, so the first question that we had is what is plant pathology and why is it important for growers?
All right, so plant pathology is the study of plant pathogens and that pathogen plant interaction. So in short, we study plant diseases, and a plant disease is actually the reaction of the plant as a response to a pathogen. So we don't study fungi, for instance, or plant pathogens in silo. We study that interaction between plants and the pathogens that cause disease. So, and we say plant pathology because that's all pathogens. That can be fungi, bacteria, water molds, nematodes, viruses. So there are a lot of different plant pathogens. So it collectively catches all of those.
Okay. Follow up to that, actually, because just this is playing on my own curiosity. And so, because you talked about things like how it reacts with the plant, right? So this is considering things like type of crop or like the genetics of that specific plant, as well as the entire climate and the plant environment, correct?
That is correct. So, so a little bit of quick terminology. When we use the word disease, that's the reaction. So if we talk about wilt, or we talk about dieback, or we talk about blight, that's disease. That's the reaction. So if you think about it in medical terms, if you've got a fever, that's not a diagnosis, that's a symptom. So we can look at that symptom, and then we have to work backwards and find the cause. And so when we talk about plant disease, we have to remember that some of the terminology and some of the categories that we loop it in is absolutely just the reaction and the descriptor. So we have to go further than that.
Oh, that's really cool. Okay,
Yeah.
especially with the medical comparison, because I mean, fever, I mean, even if we talk about COVID or the flu or whatever, you know, fever can be a symptom of so many different diseases.
That's exactly right.
And so that's really cool. I appreciate you explaining it that way.
Yeah. Now we're in COVID, and we can make a lot of comparisons that probably weren't relevant before that. So
very true. Very true. Especially understanding how like epidemiology happens, right? Of understanding, okay, the spore of, you know, some spore of some disease, how it like then, you know, replicates and creates mass.
Yeah.
And
so talk about
the greenhouse.
Hygiene, washing your hands, wearing your mask, all that we've been preaching this for years. It's applying now.
That's very applicable. But also, so moving on to the second question. So how do you support greenhouse growers and the ornamentals industry?
Okay, so I'm an extension specialist, and so whereas my position is not research, it is extension. Extension means I extend the university to the people. So I translate complicated research, and I translate that to growers or to homeowners or whoever that end user is. That includes county agents. So here in Kentucky, we have a traditional system of county agents. And so all of our counties, all 120 counties have ag agents. So my primary stakeholder is the agent, and the agent, it has the boots on the ground at every location, and a grower can call their county agent and really work one-on-one with that agent as needed. And of course, with specialty crops, it's not that easy. So a lot of times, so I do make field visits or greenhouse visits, nursery visits. So extension is my job, and I am, so I cover a lot of commodities. I cover all ornamentals. That's greenhouse, that's nursery, that's landscape, commercial landscape, and the home landscape. And even though I don't make home landscape visits, I do work with county agents, especially as we approach unusual things or things that are a little bit more complicated. And then, of course, I have other commodities that I'm responsible for. I do all fruit, small fruit and tree fruit, both commercial and residential. And I do forest trees, Christmas trees, and I do hemp. So I have a lot of commodities. And so it takes a lot of time to read all of the literature and to really know what goes on. And then, of course, as you can tell with all of these assignments, I cannot do all of the research that I need to get the data to answer the specific questions. So I do read a lot of the literature from my colleagues in other states and other regions, and I have to adapt what I read to, for instance, Kentucky's climate, or as I know, maybe a specific grower or a specific cropping system. So that being said, I don't have research responsibilities, but I do have 2 research programs. One is on hemp and one is on apple. So neither one of them fall into the ornamentals category, but I do work a lot with ornamentals just simply because ornamentals are, keep everyone interested, and they're more visible a lot of times. And the variation in ornamentals, I mean, you know that, and every year they change. So it keeps us on our toes.
Absolutely. I mean, yeah, well, especially, yeah, no, I have nothing to elaborate, but yes. So, okay, cool. Our third question is, what is your philosophy when it comes to disease management, specifically in the greenhouse, and how did you develop that philosophy?
Oh, that's my favorite question.
Good.
So we were just talking about COVID and I guess everyone, the general public's understanding of disease agents at this point. And it's a very same thing when you get into, and your question was greenhouse, is that we have to let the pathogen in or it's not there, right? And then once it's there, then we can think about hygiene, and we call that sanitation, or, you know, when it comes to a greenhouse system. So there are all these common sense approaches before we start, you know, thinking about any other way. And with a greenhouse environment, so think hospital, it's pretty sterile, right? We don't have the diversity, we don't have the biodiversity, we don't have the antagonists and the microbes, right? So we start with a soilless media. We don't even have media. And then we have artificial air a lot of times. If we don't have the air exchange, we've got, so we've got almost an artificial system, but we're not in a bubble. So whatever comes in is pretty much free to do whatever it wants to do.
Yeah.
That being said, in a greenhouse, we monoculture in greenhouses a lot of times, unless it's an, you know, an unusual situation. A lot of times we have a whole house or a whole block of, say, petunias or, say, hemp or whatever it is. So we're monoculturing, and then we're in a soilless media. So we're artificially introducing all of our nutrients. We introduce everything. So when we introduce Pythium that will cause a root rot, there are no antagonists. So my approach is common sense. Just be clean, and sanitation is my soapbox. That is absolutely my soapbox. You just have to keep it clean. You have to keep it out, and if you do introduce it, you have to get it out. And that may be pruning. That may be just roguing plants, so just throwing away plants so that spread does not occur. And I guess my philosophy behind that stemmed from just really studying pathogen life cycles. If you really know how it works, and that's what we do as scientists, knowing how it works, you could say, hey, I can break that cycle so easily. And we'll probably take this on with another question, but if viewers remember anything, is that fungicides don't cure disease. Once disease occurs, it's too late. And so we use fungicides as preventatives, right? So we can use fungicides to prevent, but then we have to do, whether, if it's a leaf disease, for instance, we can usually cut back. But if it's a root disease, we have to throw it away. So we can take all of these steps and all of this just general common sense knowledge and break the cycle, whatever that cycle is. And there are a lot of pathogens out there and a lot of cropping systems. So we have to put all of that info together and figure out how to break the cycle.
Okay, that's really interesting. Well, one of given your focus on clean, because then I'm like, because you know, I do have a little bit of background in working in a greenhouse, and I'm like, it's one of half a gajillion places that a disease could come in. Birds come in the greenhouse every once in a while. You could have pathogens on the seeds, in the substrate, you know, I mean, it's just like, there's just so many different places that's almost dang near impossible. But I mean, I do certainly agree with you a lot of just like being as clean as humanly possible, like a hospital, is definitely something.
So the number one way, so think about the most severe diseases that we see. They're typically soil-borne diseases, right?
Okay.
Like root rot, fusarium, foot rot, rhizoctonia. So think about the pathogens that we see most in greenhouses. Those are soil-borne pathogens. Mm-hmm. So how is a soil-borne pathogen going to move? In soil. Where is soil? Bottoms of our shoes, dragging along with a dirty hose on the ground, in pots that weren't washed, flats, underneath benches that aren't disinfested. So think about movement with soil. Think about your cart or your wheelbarrow that you drag up and down all the time, right? So in those wheels, in those tires. And as we talk about nurseries, it's the same thing. It's just a bigger system. Mm-hmm. You're moving any way you can move a soil particle or a soilless particle in terms of greenhouses, you can move those pathogen propagules. By the way, so tip, a propagule just means whatever that pathogen is using to propagate itself. So sometimes it's a spore, sometimes it's a sclerotium, sometimes it's a pycnidium. So the techie part doesn't matter. It's just whatever that propagule is that pathogen uses to move. And a lot of times that's some kind of survival structure that's going to be resilient enough to hitchhike for days, weeks, months, years, and just hang out until you give it, I call it the all-you-can-eat buffet, right? So you introduce it to that sterile environment with no competitors, and it's got that petunia that it loves so much.
Right. And just to clarify, right, like, because, you know, we've done some research like about botrytis and things like that. And so it's like the spore may be there and it may not cause any problems during the plant life cycle until you have, you know, those high humidities and low temperatures, which like you just said, like it creates that perfect environment. And so as a grower, understanding that that's what triggers botrytis, you could help prevent, like cleaning plus, you know, adequate management of that environment.
Botrytis is a different one. So, so my example a second ago was soil-borne pathogens, right? So those are that live in the soil, those infect roots, crowns. Botrytis is an airborne pathogen. So botrytis is what we call ubiquitous. It's everywhere. It's everywhere all the time, and it infects anything. So the one thing though about botrytis is that it's not aggressive. It's opportunistic, right? So it hangs out until you give it that all-you-can-eat buffet. And so botrytis is not aggressive enough to infect healthy tissue. It needs something that it can easily consume. So flower petals that are spent in terms of fruit, if a grower has fruit. So fruit that is overripe, leaves that are senescing, right? So it's looking for something that's already weak, and then it likes humidity. So we could talk about conditions. So you've got that pathogen that's there in high numbers most of the time. And when you give it the environment that it needs, it multiplies very fast. It produces a huge number of spores. And so it's just, it's just absolutely exponentially multiplying in the conditions that it needs.
That's scary, especially if it is everywhere all the time. Oh, that's a good reason to keep things clean.
So the time that a botrytis spore infects a leaf, for example, so you can have that one spore producing thousands of spores within a week. Thousands. So, and no spore comes alone, right? Every spore comes in thousands. So again, exponential multiplication, and very fast. So we think about insect pests that might have a 30-day life cycle or a 60-day life cycle, right? You deal with plant pathogens. Not only are they microscopic and you don't see them multiplying, but they multiply at usually sometimes 5-day, 7-day, 10-day life cycles. That's fast. That's really fast. I mean, we can't take notes and get a spray schedule going that fast. So we have to put all of our tools together. We can't just say, oh, fungicides. So we have to put all of the tools in our toolbox to work for us so that we can manage diseases.
Just a quick question here. We were talking about analogies and how the greenhouse is like the hospital and we need to keep it very nice and sanitized. If we're going back to this whole analogy and what we're in with whole COVID and talking about airborne diseases, is there an analogy to say we use masks as humans? Is there some things that plants can put on like a mask that will protect them?
That would be, oh gosh, that's a really good question.
That is
That could be fungicides, preventative fungicides, right? So it would be a protectant. It could be a vigorous plant, right? It could be a plant that's so healthy that it doesn't have the wound to get in. It's got a really a nice thick cuticle, it's rigorous, and it's got the immune system. So maybe not just the mask, but the immune system too. So even, you know, if it does become infected, it could wall it off, or it could shed that leaf really quickly, or whatever it is. So, and I was actually on a virtual field trip today with a group, and we were talking about different resistance, right? Vertical resistance versus lateral resistance in plants. But resistance comes in terms of health as well, right? So a thicker cuticle on a leaf is harder for a fungal spore to penetrate. So just simple things. Wounding, I can't say enough about wounding as well, right, Dan? Because think about a tiny fungal spore. Most people listening to this have never seen a fungal spore, right? Because it's microscopic. How is that penetrating the bark of a tree? How is that penetrating a waxy cuticle of a holly? How is that happening? It's not. It's got to get in, right? Something's got to get in there. It could be a sandstorm or a hailstorm or a bird peck or an insect, sun scald, winter damage. We could go on forever. Mechanical damage. So we are the best as humans at damaging our plants and letting our fungi in.
Can you talk a little bit more about insects and wounding? Because you mentioned that just now, and I think that's a really interesting point because they do pierce wounds into plants.
So if we're still talking about the greenhouse environment, we know there are a lot of insects or arthropods in greenhouses, right? So think about thrips. So thrips, when they bite and they rasp, so they just jerk. And so they're tearing plant tissue, right? Then you've got aphids. They are sucking insects. So they probe and mites as well. And they suck the contents out of a plant cell. So that's all damage. And then of course there's the weakening, right? Because when you don't have your intact leaf or a healthy leaf, you're not photosynthesizing, right? Or you're disrupting uptake of nutrients and water. So you're not just stressing plants and weakening plants, but you're actually creating wounds. If we were to talk about the landscape outside, so think about woody plants. I mean, they're perennial. So what you do today is going to continue to affect, let's say, a tree or a shrub years to come. Because plants don't heal, right? They can wall off in certain circumstances, and other times that wound just kind of waits to callus off, and that takes a very long time. So you've got an open wound. So think about us. Think about a wound on a human body. How long does it take to really heal, and what infection can set in the meantime, right? We don't have Band-Aids for trees. We don't have Band-Aids for every cell that a spider mite penetrates, and there's never a spider mite, right? There are millions of spider mites, not millions, or there may be, but these small arthropods don't come in one or two like a Japanese beetle. They come in very high numbers, so they're doing a lot of damage to a lot of cells, making a lot of wounds, and then those opportunistic plant pathogens come in.
That's a really interesting perspective. Thank you. Appreciate that. Moving on to the next question is, so you talked a little bit about in the first question of, you know, how your role within the entire, you know, in the extension system. And so, but when someone comes to you with a disease problem, what is the process that you guys lead them through?
Oh, okay. So I consider myself a field pathologist. I'm not just the microscope, give me one leaf. So
okay,
I like to be on site. I like to see the whole site. And that's when you see the insects, the poor drainage, the dirty greenhouse, you see all those things. But so what I need to figure out, I can always find a pathogen, right? Dan, right? We're plant pathologists. We can always find disease, like it never fails. And so we have to ask the question, is it a primary disease or is it a secondary disease? That's the $1 million question, because there will always be disease. So what's the real problem? And what I have found in ornamentals primarily is that about 2/3 of the time, there's no pathogen anyway. It's abiotic. And then probably only 10% of the time is there a primary pathogen, right? So even if I can find a pathogen, if I can find disease, a lot of time there's something else going on. So when you call me, I have a laundry list of questions. I want to know everything. I want to know your fertilizer schedule. I want to know your herbicide schedule. I want to know what the temperatures have been. If it's a greenhouse, I want to know, like, what's your data logger look like? Do you have what's the humidity in your greenhouse? Mm-hmm. And all of those things really matter. It's like Sherlock Holmes. You have to have all the clues. And sometimes you never really get to what the problem is, but I can tell you what it's not. And you need to help me figure out what it's not, right? So if you are not with me, if we're on Zoom or if we're on the telephone or even exchanging photos by email, you have to help me see patterns. You have to help me see all of those things so we can say, okay, it was a temperature, or the drainage is okay, or your fertility is up to par, whatever it is. So I'm going to run you through an approach for eliminating all the very common things. And then from there, I need information. And sometimes I'll ask you the same thing more than once, or I'll have an idea, like maybe it's, something odd. And so it's just kind of going through. And two, we're always in tune to what's new and invasive that's coming across our borders. And so to be able to know that, and sometimes things just ring a bell. So basically, and I can't go in with only my plant disease bias. And of course I do. And I've got to back myself up and say, okay, do we see any insect frass? Do we see any patterns? Like, what do the roots look like? So if it's a container grown plant, I'm going to have you pull it out. I want to see what the roots look like. And all of those things come into play. So it's not as simple as these leaves are brown. Here's a picture of a leaf. What's wrong? I wish it was that easy, but it's never that easy, right?
So then follow up to that of I could make some assumptions, but assumptions get me in trouble. And so could you elaborate? Could you elaborate on a little bit more, like what are examples of primary? You said primary diseases, and what are examples of like secondary diseases? Okay,
so a primary disease might be Pythium root rot, right? So that's an aggressive pathogen, and it just needs a host and water, and it can do its thing. So that's one that would be a primary disease. Now, once you have Pythium root rot, you're surely going to have nutrient deficiency, because without roots, you can't have the uptake. Right? And then when you have that weak plant, there are a lot of arthropod or insect pests that are ready to come in. And surely botrytis will be really happy to see a declining plant. So Pythium root rot might be a very good example of a primary pathogen. It's aggressive.
Okay.
Rhizoctonia is aggressive. The fusariums are aggressive. And then we can look at like the landscape. So, and landscapes are really a prime location to look at secondary or even tertiary diseases. So in Kentucky, for example, we're on the cusp. We're a transition zone between the north and the south. So we have hot summers and we have cold winters. One of the things that happens to us, so we're cool enough that everyone likes to plant spruce and some of these upland species, for example. But at the same time, we get so hot in the summertime. Well, we push our limits and we plant things like, crepe myrtle that we shouldn't have. So what happens in Kentucky is usually at the end of February or beginning of March, we'll start seeing some warm days and our plants break dormancy. And then we get another freeze. And I've seen the coldest weather in March, right? So we'll have a 70-degree day and a 10-degree night, all within a week or two from each other. And so once plants break dormancy, they are susceptible to cold damage. So that's what winter injury means in my world, in the world that I live in here. And then also that this very same things can happen. We can have a 90-degree day in October, and we can have a hard freeze in November, and plants haven't hardened off. So when we have that damage of a cold damage to a plant, you can call it spring damage or winter damage. It doesn't matter what you call it, but it's not a dormant plant that's damaged. It's the plant that is not dormant that has the cold damage. And so with that, you have plant death, right? You have dead twigs and, you know, those types of things. But it's the sublethal damage. So it's the damage, not death, that the plant is still technically alive or the branch is still alive or whatever that may be. And then you've got open wounds, you've got freeze cracks, you've got, you know, this damaged tissue. So you have rhizosphaera needle cast on spruce. We've got a lot of our cankers like Cytospora canker. There are a lot of cankering diseases that will occur in woody plants. Boxwood, oh my gosh, so boxwood, very, very disease prone for us, and it's winter damage. And then fall blight comes in, and then of course that's where the blighting, the straw colored leaves that remain intact, and it'll usually be on one side of the plant, like maybe where there's morning sun, where that plant broke dormancy, you know, most readily. So those would be secondary diseases that kind of came in that I would blame an abiotic primary cause. And that's again, and that's, you know, it's pretty debatable, you know, what's primary and what's secondary. And as a field pathologist, that's my version. If you submit that sample to a plant disease diagnostic lab, they're going to report exactly what they see. They don't see the site, they don't see the history, they don't see the information that's not on the form. And so it might be listed as a primary disease from that standpoint. From my standpoint as a field pathologist or to work with the county agent, it's not. So it's kind of, it's kind of dependent on who you talk to.
Okay, well that still provides a lot of great clarification, so I do appreciate that explanation.
No disease exists in a bubble, so there's always several other things at play.
I mean, and lengthening the medical, you know, analogy we've got here of like, you know, someone can have a disease, but then it can develop into pneumonia, and then it can, you know, bronchitis and like things like that of like, what are the subsequent effects that can happen to humans? Okay. The next question that we had was, what is the most common mistakes that growers make when managing their greenhouse in regards to plant pathology?
Sanitation. You know, we started out, you know, when we arranged this conversation, you know, sanitation was kind of at the cusp of what we were going to talk about. And you can't get away from that. It's sanitation. Just keep it clean. You know, in a greenhouse, don't introduce disease, don't introduce plant pathogens, and keep your plants healthy. And in terms of introduction, we can talk about quarantining and just, you know, not just introducing new plants into a house that's already healthy, you know, because that risks introducing new pathogens and contaminating everything in there. But that's probably number one, and not fallowing a house in between crops. You've got to clean it out. You've got to sanitize so that you can, any propagules that are lingering in those soil particles or you know, in the you know, in the cooling pad, that you can sanitize, flush your lines. So fallowing the greenhouse, pet plants, right? Are we all guilty of bringing our tropical plants? Well, maybe not you, you're in Florida, but bringing our tropical plants into the greenhouse to make the winter, and, you know, bringing all those hitchhikers in. We're all guilty. I do it. Like I tell everyone not to do it. So confession, I do it.
Yeah.
So, you know, I had some elephant ears in my bedroom last winter, and I had like alcohol and cotton balls, and I was getting the scales off, you know.
Yeah.
So we do those things, but don't do as I do. Do as I say do.
Yeah.
So
well, I can just imagine of things like, you know, trying to think of, because for the record, so I actually live in Illinois. I'm from Florida, but I'm currently in Illinois.
Okay.
It's just where I've decided to live. And so I will do that because, I mean, it's end of August. And what's really terrible as a native Floridian, I don't understand this thing called fall and winter. And so you laugh, it's a problem.
I'm guilty.
So last winter, we had a snow. The first snow was October 31st on Halloween. I had trick-or-treaters, you know, Cinderella was in a heavy down coat. And so I was thinking through this recently, and I was like, so you're telling me that I could potentially have snow on the ground in 2 months? Like, that's a very depressing thought.
Yeah,
first.
Yeah, that's right.
Second is with this same sort of deal, it's actually something that I've thought about, is like, because I have plants inside. I have, you know, we're doing an internal chili challenge of like who can grow the best chili, the most chilies, things like that. And I'm like, well, dang, like I have plants outside. I'm getting ready like within the month to bring them inside. Like, what if I bring in some sort of, you know, pest or disease and it attacks my pepper plant in the chili challenge? I'm like, am I going to jeopardize, you know, that growth of that plant? It's really unfortunate.
And you know, we're homeowners with these hobby plants, but think about a commercial greenhouse.
Exactly.
And a brother-in-law says, oh, can you put my palm in your greenhouse for the winter? Because it's so, you know, so you do bring other people's pet plants into the greenhouse, or you introduce, I'm going to save this mother plant. I'm going to take cuttings on this in February. So yeah, we do it.
And not even that, of even just thinking through different customers that I've talked to at a commercial scale, and like the amount of sanitation process and like verification of quality, right? Because, you know, as like tomato, pepper, cucumber growers, I mean, you're like assuming they're hectares and hectares and hectares of greenhouses. I mean, you're changing thousands of plants, having that changeover in a matter of days. And so like how, like that process of getting everything out, sanitizing, assuming that they're really not letting the greenhouse fallow very much before they then put, you know, those thousands of plants back. I mean, having everything in that process laid out and very concrete has to be very, very important, particularly as we talk about plant pathogens.
And it does. And you have to be ready. You have to have everything there, and you have to just demand that your next crop is worth a couple of days, you know, or whatever that schedule is. You really have to do that, because if you have one outbreak, so again, fungicides don't cure disease. Once disease sets in, you're chasing your tail. So to be able to have a plan and to really disinfest, to sanitize, and then to watch carefully as plants move in, and be careful about introducing new plants, right? And again, not just hosing down the sidewalks, to really, you know, think about, do you have wooden surfaces on your benches? If that's the case, bleach is about the only thing that will disinfest wood. We have some other products that will get hard surfaces. You've got to flush your lines. You've got to clean your tools. I mean, all of those things. Reusing pots. You know, if we have a disease outbreak, we always want to save the pots, right? And, you know, I mean, I really am very conscious of plastic. So I reuse as much as I can. And smaller businesses will reuse. You've got to clean them. You really have to clean them. You can't just shake out, you know, soil that's riddled with Pythium propagules and reuse that, because you're reintroducing.
Yeah, that's definitely the big challenge.
Yeah.
In the essence of time, I'll go to the next question. So how should someone efficiently and effectively scout for plant diseases? Because like you said, I mean, you know, we talked about introducing it, things like that, keeping it clean, things like that. But like you said, things like botrytis, and I'm sure a lot of other plant diseases are already there. And so how do you scout for that?
Okay, so you're looking for patterns, or whether there are patterns or whether there aren't patterns. So you're looking for patterns. You cannot scout every plant in a large facility, for example. You can't scout as often as I would like you to, right? Again, you know, I can tell you ideally what to do. So let's backtrack a little bit, and maybe we should have talked about in the very beginning, is that plant pathogens need two things to complete their life cycles, to cause disease. They have to have a susceptible host. That could be damaged, that could be a, you know, something that's maybe susceptible. Sometimes it's species, sometimes it's tissue, like for instance, botrytis needs something that's decaying or in decline. Pythium needs a root. Some pathogens will infect only new tissues, some will only affect blossoms, etc. So you need a susceptible host or plant part. And the second thing is you have to have a conducive environment, and that's moderate temperatures and free water. Moderate temperatures mean if you're comfortable, that pathogen is comfortable. That's why we don't see disease issues in January, and we don't see disease issues much in August because it's either too hot or too cold. So you have to have moderate temperatures. So I like to say, if you're comfortable, so is your pathogen. And we get that a lot in springtime. We get that in a shade house. We get that in microclimates. And then we need free water. Free water means any humidity level above 70%. That means irrigation. It means rain. It means fog. It means dew. It means a lot of things. It means a wet soil that's causing the inner canopy of a planting to have high humidity inside that canopy. So you need free water. And so back to the scouting. So if you're going to go into a large nursery or a landscape or a greenhouse, where would those conditions be? And because you have a higher risk for seeing disease or having the earliest disease in those areas. So if it's a landscape, you can look at the low-lying areas. You can look at the north side of a building where the sun never hits. You can look at where the gutter spout comes out. If it's a greenhouse, you know, the sun still comes up and down, right? So you still have morning sun that dries leaves off really quickly, and you have less of that dew sitting on leaves. Or you might look at maybe near your, like if you have one bench in your greenhouse that you've got your misters on, or you're watering more, you've got more water there. So think about where you might see the first signs of disease, more susceptible plants, etc. So look for potential patterns. So you can look at the most likely places to see disease. You can also start just standing back and look as a whole. What's the overall color of the plants? Is that right? What is the, you know, do you have maybe on the far edges, maybe on the west side or the south side of a greenhouse, things might look a little bit more brown. So start looking at patterns. Overall, plant pathogens don't have patterns. Abiotic or environmental conditions, you will see patterns. Overall, that's just general, that's not set in stone. At the same time, look at your cultivars, right? You might have one greenhouse bench of petunias that's, you know, a certain cultivar. They're all affected or mostly affected by a problem. And then you might have another that's not. What's the common denominator? You can scout a plant, a single plant, the very same way. Look at the whole plant. What's the common denominator? Is it just one branch? Is it just one tip? Is it the whole thing? Is it, you know, so, and when you look at the common denominator, it might just be a small branch. It might be a, you know, the trunk or the crown or the root. So think about it in patterns across a landscape, patterns across a greenhouse, patterns across a block in a greenhouse or a block in a nursery, and then patterns within a single plant.
Okay. And so I think, sorry, say that one more time.
I said, and then take notes because you'll forget. And when you call, I'm going to ask you those very questions.
Right. It sounded very familiar of what you had said of like, of walking through the greenhouse and being on site as a field pathologist. So yes, right. No, that is where my brain went a little bit. I think that segues very nicely, because you talked a little bit about, you know, one greenhouse versus one side versus the other, or you know, different variations across the entire greenhouse. So how can understanding the microclimates within the climate help prevent plant disease?
It's very much the same thing. So think about, where is that free water? Where's the highest humidity? Where is the shade? Because in the shade, you have a slower drying, right? The microclimate could be where a dripping irrigation spigot. It might be where the cooling pad is. It might be on the north side where the sun never really comes through. It might be under a bench. You know, we're all guilty. We're back to greenhouse analogies, but are we all guilty of putting plants underneath the bench? You know, and they've got secondhand water and contamination. So think about what's happening. Think about the weather or the climate. So a microclimate is just a small version of that. But you know, and even if we're thinking like landscape nursery, we think, but it's been raining. Well, it might not be raining underneath an oak tree. You know, that water might not penetrate down. Or, you know, it's been very dry. Well, not where, you know, the neighbor's pool overflow is. I've seen those kinds of things. So think about anything that could be different from the norm. And we're not lucky enough to say the weather is the weather is the weather. We've all got microclimates everywhere. Even in the most consistent situations, there will be microclimates. The inner canopy of a plant is a microclimate compared to the whole. They're always microclimates.
Exactly. It's just understanding how that plays into when analyzing, like you said, all the patterns. So I really appreciate you saying that. We're almost done, so 3 more questions. So do you mind going through a specific greenhouse disease that you have worked with, and what are some red flags that growers should look out for?
Oh, a specific disease. I've used Pythium several times. So let's talk about mums.
Okay.
So a lot of mum growers here and in a lot of places. So mums or chrysanthemums are grown for fall sales. So they're generally here, they're planted in June and they are sold in September, October. And so mums are grown on a nursery pad, like a nursery crop, but they've got drip irrigation just like a greenhouse. So it's a hybrid system and they're in containers, they're spaced far apart. And we've got more and more mum growers here in Kentucky. And I know in surrounding states, very much the same thing. And so mum growers are often either new to mums or they are new to this type of system. And so I'll get calls in and say, I'm having problems with my mums. And to really look through a site. And so what we're finding is, so there are 3 major diseases of mums. That's Rhizoctonia web blight. So web blight, Fusarium wilt, so that'll be a dieback, and then Pythium root rot. And a lot of times the symptoms will be very similar. They're all soil-borne pathogens. So what I'm seeing more and more, and this is not one site, this is very common, is that so when growers set this up, they put out thousands of mums. They'll stretch a nursery cloth, a ground cover cloth, down on the ground, they'll pin it down, and they'll run drip irrigation. Everything goes through the irrigation. First year, they usually do very well. In fact, well enough that they expand that planting. The next year, they'll start seeing some disease. And they'll see 1 of the 3 diseases, 2 of the 3 diseases. The next year, there's even more disease. So it accumulates. So what's happening is these soil-borne pathogens are building up in the soil underneath that ground cloth. And so think about when you're watering, so the water goes through the pot and it goes through the fabric and then into the soil. Well, then when you have a lot of rain, so think about like when you pick up a plant, it's wet underneath. So you have a water exchange, water and soil exchange. Right? And even things like fungus gnats, they transmit Pythium. They carry Pythium spores with them. So you have this happening. And so what we've started recommending, and this is expensive, but for growers to take that cloth out and put about 4 inches of rock and then put that cloth back down. So you've
Okay.
now created a barrier between the soil and that container. Okay, and then we also are recommending for some kind of disinfestation, so some kind of sanitizing product. And I do have a new publication I will link for you with disinfesting greenhouses, but this works for the nursery cloth as well. So to disinfest it and then to flush those lines. I talked earlier about flushing lines. So that my example is that buildup of those soil-borne pathogens. First you're nothing, second you're a little, and just more and more every year. And that really can devastate, you know, when a grower loses 20% of their mom crop or 10%, that's the profit margin. That's huge, right? And then it's illegal to sell a diseased plant. So once you know what you've got, then, you know, can you throw away 5,000 mums at $10 a piece or $5 a piece, even for the small ones? So it can be really devastating, but just to understand that concept of buildup of soil-borne pathogens is really kind of eye-opening sometimes. If it was mine, I could lift them up and just, you know, put some kind of, you know, bricks or, you know, inverted trays. But when you've got 5,000 mums, it's not so easy, right? So you really have to do something that's efficient and effective and cost effective. So that's an example that we see a lot. Leaf diseases are easy. When you have root and stem diseases, that plant is toast. That plant has to be destroyed. And that's much harder than a leaf spot. You can cut back the plant and it's rejuvenated and you can salvage it. You can't salvage a root rot or a stem rot in a perennial or an annual crop.
Yeah, I mean, even when I worked in the greenhouse, when we got hits of Pythium, I mean, you're done. You can't do anything. And this was hydroponically, right? So we were in water. And so it's just, no, and even I like that example because that brought up something we really haven't talked about, was the accumulation of things over time, of certain pathogens over time, and how it's not even just of like you get hit one year and that's it and you're done, and maybe you spray and like pray it's not going to come back, but like chances are like the accumulation over time, like I definitely appreciate that example.
So our plant pathogens produce almost all of them produce survival structures, some type of structure that the pathogen can live outside of its host. And a lot of times those structures might be melanized, which means they're UV resistant, they're drought tolerant, they're cold tolerant, they're heat tolerant, and they can really survive. They can either overwinter or over summer with, you know, in this propagule form. And so, you know, if we wait until there's disease, or if we don't rogue quickly enough, and when that pathogen goes into survival mode, it's shut down, it's locked down. It can tolerate almost anything. And it's just waiting for the next crop. Yeah, no, that's definitely lots of things to think about. And I have learned a lot in this conversation. So the last question that I was going to ask is, so what do you recommend for next steps of growers for them to do? Maybe you've sparked an interest of like, hey, I have that, or maybe they realize they need to look into something and have somebody come out to their, like, what are the next steps that you would recommend? Okay, so you have a wide audience here, right? Mm-hmm. And so what I recommend for my growers will not necessarily be what's recommended for every grower. So go to your cooperative extension in your state. There will be websites, there will be publications, and read what they've got. So any data that you find or any recommendations that you find, I highly recommend that they come from a university source. And that's because we have nothing to sell, right? So we're presenting an unbiased approach. So that's of course, as a university scientist, that's what I recommend. So start there, and we'll do the nerdy kind of publications. We'll tell you about the life cycle of, you know, whatever, botrytis. So we'll tell you about the life cycle. We'll tell you how it overwinters and oversummers. So get some actual data, some information from a reliable source. And in our world of social media and everybody can have a website, there's so much bad information out there. Mm-hmm. So start there. And then get to know your county extension agent or your county or your extension specialist. So get to know the people that you're working with and give as much information as you can. We were talking about greenhouses the most of today, but how big is your greenhouse? How many crops are in a single greenhouse? That makes a difference in how we make recommendations. And then secondly, if you are working with a company, whether it's a fungicide company or a greenhouse manufacturer or whatever, ask questions, right? Because it's very easy to trust others, especially when it's a complex topic, but you really have to know what's going on. You have to be able to ask questions and understand. And if you don't understand, say, I don't understand. And then they can explain it a different way. But just be prepared that fungi, or not just fungi, but plant pathogens are complex, they're microscopic, and they really play tricks on us. So it's hard, but management has to start early, and you have to trust whoever it is you're working with. You have to trust that plant pathologist, or if you're working with an entomologist, you have to trust that entomologist. And you have to just stick with it. It's like, again, a medical analogy. When you go to the doctor and that first recommendation doesn't work, you've got to go back, right? You have to keep going back until you tweak that solution, whatever it is. And we're doing the same thing. So you really have to educate yourself and make sure you've got the right information. But I highly recommend working with your extension service and be careful about looking at recommendations from other states. Sometimes it applies, sometimes it doesn't. If you're not sure, just ask your county office. Just say, does this apply here too? Like if, for example, if you had one of my Kentucky fact sheets, you say, does this apply here for me in Illinois or in Florida? And they may say no, and that's okay. It doesn't mean the information's wrong. It means it's not applicable to you and your situation.
Right. No, definitely. So that is all the questions I have. And so we do really appreciate the time and the information. I certainly learned quite a lot. And it's been really fun working with Dan and the rest of the Folium team, as well as then thank you so much for the time here.
Thank you, Dr. Gautier.
Yeah, good to meet you, Dan. Good to meet you, Kayla. And anytime you have questions about plant disease, I'm a talker. So I'm always happy to talk about disease. And if you need any other topics, let me know.
Absolutely. We appreciate it.
All righty. Bye, everybody.
Nice to meet you.
Bye,
See ya.
guys.